Hey Beaklings, it's Mr. Boylan. We're going to kick off Unit 8 today talking about cell division. Okay, this is a very important unit with the cell cycle and mitosis and meiosis, so we'll be covering a lot of important concepts here. And we'll start off talking about cell division and the roles that it plays. So there's two major types of cell division. If you look here, this amoeba is dividing, right? And that's just going to form a clone. Okay, that's asexual reproduction when a parent creates an identical copy of an offspring to itself. Okay, and we certainly do not do asexual reproduction. We do sexual reproduction, as do many organisms. And this is the fusion of what we call gametes or sex cells. And obviously for human, we have sperm for males and eggs for females, and when they come together, that's how we form organisms, which we'll get into later. Okay, and dogs are interesting. Obviously, that's sexual reproduction used there. And there's all different types of dog breeds, but they're all the same species. Right? Just like humans. Look at how we all look so different from each other. But obviously, we're all the same species. So sexual reproduction can lead to lots and lots of variation inside of a population. And that's actually going to be a big key later this chapter. Because we reproduce sexually, our species has a lot of variation. We're in good shape because of that. Okay. Comprende. So here you can see, you might know Mario, right, from Nintendo. This is Luigi. Okay, this is Mario's little brother right here, or taller brother. Anyway, why do the progeny or offspring here look different from their parents and fellow siblings if they're all made from the same DNA? Well, again, what you're going to learn about this chapter is sexual reproduction. You have these two parents that are randomly going to shuffle and make gametes. So even though you might have a brother or sister, maybe even from the same two people that made you, right? But because that when we make our gametes or our sex cells, we shuffle everything together before we deal them out and put them into those sex cells so they can be used for reproduction, for fertilization, we can get some really, really crazy combinations of genes, and that's why we can look different even from our own family members. Okay. So, Rudolf Virchow, right? He said that all cells, if you remember, come from pre-existing cells via cell division. So cells had to come from somewhere, sure, and if you go on to AP Bio someday, we talk a lot about, well, how did the first cell ever show up on planet Earth? And it's really cool. But basically, all cells come from other cells, and then they just keep dividing over and over and over again. So if you're a unicellular organism, sure, you're reproducing a whole organism then every time you go through cell division. And some multicellular organisms can reproduce asexually as well, like a plant growing from a cutting. Sure, you can make a clone of that. You do the same thing, right? You fall and scrape your knee or something like that. Sure, okay, you're going to be doing asexual reproduction there too. So your cells can do asexual reproduction if they're upkeep or growing or something like that, but we don't create new organisms, okay, via asexual reproduction. We need sexual reproduction for that. All right. And then we have sexually reproducing organisms start cell division right after fertilization, which is going to be important later. And new cells come from old cells for the rest of the organism's life. Okay. So, comprende. Starting with a fertilized egg, a series of 5 cell divisions would produce an early embryo with how many cells? So, fertilized egg is 1, right? And then you divide into 2, and then 4, and then 8, 16, and okay, 32, right? So hopefully that makes sense. And here you can see an urchin starting its life here, which is pretty cool. Let's speed through this here. You can see cell division, right? Blastula of all the cells, and then start making all the good stuff on the inside of that. Pretty cool. Okay, so I have a good story about one of these little urchins here when I was in Hawaii. So next time you see me, you can ask me about that. It involves lava and curses, and it was crazy. Anyway, prokaryotes produced by binary fission. Okay, now this is super simple. We're going to have to spend a lot of time later talking about how we copy our cells because it's really complex. So we're going to do that in the next video. But for today, we can talk about bacteria because it's pretty easy. So here's a prokaryote, right? And as you remember, they have a nucleoid region where their DNA is. Basically, they do binary fission. You just split in half. You copy your DNA and split in half, and you're good to go. Okay? They usually just have one huge chromosome. It's a big ring, all right? And when elongated, the DNA of E. coli is around 500 times the size of the cell. So we're going to talk about how we do have to have a way, especially eukaryotes, to organize all of this so that we can do it efficiently and correctly. So again, basically, what do prokaryotes have to do here? Well, they duplicate their DNA. Okay, here's their single chromosome, big ring, and you have 46 linear pieces of DNA. They just have one ring of DNA. And they copy that, they elongate the cell, okay, and then plasma membrane grows in between them, and then they also have this cell wall on the outside. So why would binary fission be considered asexual reproduction? Well, as you can see here, there's offspring are genetically identical to the parent, right? That's why a lot of times these antibiotic soaps and things like that, if you think about it, you go ahead and clean your hands. If you're going, which right now we're dealing with the COVID virus, right? But if you wash your hands really well with a certain antibacterial soap, which we probably shouldn't be using, to be honest. We use way too many antibiotics in our normal lives. But why does it kill almost everything? Well, think about it. If these bacteria are all from the same line, right, they're all clones of each other. If you can get one, you could probably get all of them. We'll talk about ways later that bacteria can change themselves up. But asexual reproduction, you're basically just a clone. Okay. So we have the large complex chromosomes of eukaryotes duplicate with each cell division. All right. Your cells are way more complex, as you know, than prokaryotic cells. We're larger and we have way more genes, theoretically. Now, this text that I use says 30,000 genes. It's probably a bit less than that. We're probably over 20,000, under 30,000, somewhere in there, but bacteria certainly less than we have. Our genes are grouped into many chromosomes in a nuclei. Okay, and we do have some of the mitochondria as well, which is important. So I know we've been using those words a lot this year already, chromosomes and chromatin and things like that. It's time to figure out what they mean now, this chapter. So chromosomes normally exist as a fibrous mass of DNA and protein called chromatin. Basically, you all know that the double helix is your DNA molecule, right, inside of your cell. Well, the double helix is one of your macromolecules, right? It's sugar phosphate bases, hydrogen bonded together, these polynucleotides. That's the double helix. But generally how it exists in your cell is that DNA wraps around other proteins, these little histone guys. So instead of just having a double helix, you're actually going to have a thicker thread of a lot of DNA wrapped around other proteins. That's what we call chromatin. So I like to think of it as if DNA, the double helix molecule, is like fishing string, thin fishing string, then maybe the chromatin fiber is like a real thick spaghetti noodle or something like that. Okay, so if you look here, we have this African blood lily. Okay, and I can tell already just by looking at this, this is ready to join into cell division, because you would not be able to see DNA like this. Okay, maybe in lab we looked at cheek cells and things like that, and you could see a purple blob. But if you actually see strands, these are chromosomes, and that means that the chromatin, those spaghetti noodle thick fibers that I was just talking about, coiled up even more so. These are gigantic, okay? These are like millions and millions of spaghetti noodles laying on top of each other if it got to this point, okay? So chromosomes only coil up when you're going to divide. And they're named colored bodies, chromosomes, because chromosomes really have an affinity for stain. And a lot of that is because of the proteins in there, and definitely DNA has that phosphorus backbone, sugar phosphate backbone, and it's charged and it loves stains. So it's easy to see the DNA a lot of times. Okay, super important here. You should know the structure of chromosomes and why chromatids form. All right, so I would just say pause the video if you have to, but spend some time really looking at this and make sure you understand this picture and what a chromosome actually is here. Okay, so as you can see, we have a chromosome. Okay, and what it is it's made out of two genetically identical sister chromatids. Now we're going to draw out mitosis soon, and I'll talk more about why these are genetically identical. But what it basically means is whatever the double helix is here, okay, that made up this side, with all the proteins and stuff mixed in, this is exactly the same A, T, C's, and G's, the exact same genes and what we're going to call alleles. So for example, if on chromosome number, 7, I don't know where the gene actually is, but if my father gave me a certain trait like earlobe attachment and he gave me free earlobes, the gene right here, well, that means that right here, the same thing would happen here. It would be a free earlobe. My earlobes would hang down because these sister chromatids are genetically identical. Now, what about if my mom gave me a chromosome? Well, if it's the same chromosome number, let's say number 7 for my mom, she also gave me the gene here at this locus and here for earlobe attachment. But maybe my mom has attached earlobes and she gave me attached earlobes. So that just means that the A, T, C's, and G's are a little bit different here. So we have a different, what we call an allele. But all that means is that the genes are the same, but the alleles can be different. Okay? And basically, these sister chromatids are held together at something called the centromere. Okay? So if I had to use my hands here, I'd say like my wrists are the centromere, right? Okay? And when and if they do come apart, okay, which we'll talk about this during mitosis and meiosis, it's very interesting. When you break the centromere, okay, and the centromere is a region of DNA that's in the center of these chromosomes, and that's where they connect. When you break these apart, you created 2 daughter chromosomes, and that's going to be very important later. Okay. Now again, you would never see this thing over here. You would never see that chromosome unless your cell was going to divide, right? Your DNA is just stringy spaghetti noodles, okay? But when it comes time to divide, no, we have to coil up. We have to make sure that these chromosomes get nice and tight and coil up because it's much easier to separate that way. Okay? All right. Well, see my computer's acting up a little bit here. The cell cycle multiplies cells. All right? And what is the whole point of cell division? Well, it allows organisms to reproduce, grow, replace worn out cells, maintain a constant number of cells. Right? We do it all the time. You clap your hands for me when I'm finished this lesson. You probably just destroyed some cells in your hands. You have to replace them, right? Or maybe you're sitting there eating pretzels right now. Are you? Are you sitting there eating pretzels right now? Well, if you were, you're probably, as you swallow them, you're scratching your inside of your throat and things like that. You're destroying cells. You're gonna have to replace them, right? So even when you're your age, we're doing this regularly. And you have about 100 trillion cells, and each cell type divides differently. Some every day, like we just said. Some take longer. Some never do. They divide early on, and they probably don't divide again for the rest of your life. Okay, depending. There's some research that says otherwise, but for the most part, some cells just stay in what we call G0. The cell cycle is an ordered sequence of events that extends from the time the daughter cell is formed from the division of a parent cell until that cell divides, forming 2 new cells. So the cell cycle is basically just saying it's the division of a cell, and now when you have this little cell, it's going to get bigger and divide again. That's a cell cycle, from the time you divide until the time you divide again. And there are 2 broad stages of cell division. We have interphase. Inter means between the main phase here, and then we have the mitotic phase. This is what we're after when we draw it out later. This is what we've talked about so far this year. Your cell is a factory. All those things going on in your cell, that's interphase. But now when it's time to divide, now we're in the M phase. Okay? So in interphase, the cell's metabolic activity is very high. The cell grows, synthesizes organelles, duplicates chromosomes, increases proteins. This interphase, and if you notice here, it's everything here, okay, except the blue part. So G1, S, and G2 are all interphase. It's 90% of the cell cycle. Okay? And yeah, that's your cell working. That's you making proteins and shipping them out from the rough ER on microtubules to the Golgi and these vesicles. And that's your mitochondria working hard. And that's your lysosomes breaking things down. Your cell is doing everything it should do during this time. Okay? There are some weird names here, though, like G1, for example. This is the first gap. Now, you just split, right? So we're starting a whole new cycle here. This cell just split, and scientists, if they're looking under a microscope, would say, oh, okay, not much I can see going on here. So it's kind of a dumb thing now we know to call it this because they called it the first gap. It's a misnomer, like a gap in activity. But you know that's not true, right? Because the gap in activity is just what you can see in a microscope, but there's a lot of stuff happening there. But then we get into the next phase, and oh boy, is this one important. Okay, S phase, or DNA synthesis, or what we're going to call the replication of DNA, and that's a really cool process that we're going to get into later this year. So S is for synthesis, is for the replication of DNA. Very, very, very important step. Okay, and you can see it takes a long time during interphase for this to happen. You have 46 chromosomes that you have to copy. Okay, that's kind of a lot. All right, and then we get into the second gap. Okay, and again here the cell's growing more. It's getting ready for divisions. It's increasing certain proteins to a threshold so that you can trigger to divide, and that's basically the end of interphase. So again, scientists lumped these three phases together and said, oh, okay, in between the time that we're actually doing this, dividing, we're just going to call that interphase. But that's the majority of the time. That's when your cells are working hard. So here we go. Now we're into the mitotic phase. So if you paid money, like you're going to a concert or something like that, and you had to sit there and wait, and you're putting up with all this other thing, and then there's this beginning cover band who's terrible, and they can't sing at all, and then you have some DJ who doesn't know how to spin a record, and all this stuff. That's kind of like interphase. Ugh. That's not what you paid for. We're here. We're ready for M phase, or the mitotic phase, right? This is only 10% of the cell cycle, but this is cool. This is where you're going to see some really neat things happening under a microscope, all right? And that's because your cell is going to divide. And you can see it's divided into two parts, mitosis and cytokinesis. Mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm, or the cell, okay? So again, this is not easy to do. Imagine that. You have 46 chromosomes inside of that nucleus, and you have to divide them exactly right. Right? Not easy to do. You're talking 3 billion base pairs. That is really challenging to do. So mitosis, very complex, and that's why we're going to draw it out together so we make sure we understand it. And then cytokinesis, not too bad for us because we're animal cells. We don't have a cell wall. We just pinch in half. But the cytoplasm is divided in two. And if you think about it, well, now that you just split a cell, where are you at? You're in G1 again, right? You're right back into interphase. Okay. So, cell division is probably an evolutionary adaptation to copy huge amounts of genetic material quickly and efficiently. Studies have shown that errors only occur about once per every 100,000 divisions. But they do happen. And technically, if you really do have, billions of bases in there, well, that means mistakes happen all the time, right? And we do have ways to fix them and check for them. But, comprende. Here we go. Oh, I gave you the answer already. Sorry, my computer's acting very funny. If a researcher treats a culture of cells with a chemical that prevents DNA synthesis from starting, which part of the cell cycle would the cells be trapped in? Well, of course it's going to be G1, right? Because if you think about it, we're in M phase, and that's when you divide. So if the researcher treated you with a chemical, okay, that prevents DNA synthesis from starting. So let me think this through here. All right. M phase is when we divide. G1 we get bigger. S phase is when we copy our DNA. Oh, so if we're treated with a chemical where we can't copy our DNA, we're stuck back in G1. Right? So that's a good example of like an honors level question that you might see on a test. Okay. And that pretty much wraps it up here today. So I hope that made sense to you. Cell division is super important, and we're going to dive into the complex parts today. This was just kind of introductory stuff, but I hope you found that helpful. Okay, I'll talk to you later.